Friends, in this video I'll explain about the JAK-SLAT pathway of cell signaling. So in the JAK-SLAT pathway, for example, if you're considering this as a cell membrane, and here these are the cytokine receptors which bind with the cytokine, and they are situated quite distant from each other, and they actually having an enzyme called Jesus kinase within their cytosolic site. These are the JAK kinase, Jesus kinase or JAK kinase. But after the binding um, of the cytokine, let me draw the picture first. Binding of cytokine, for example, this is the cytokine. Binding of cytokine to the receptor. The receptors are actually come close to each other, and so that their Jesus kinase can phosphorylate each other. This can phosphorylate and this can phosphorylate each other on their tyrosine residue, phosphorylated form of Jesus kinase. Now after that, what will happen is that, let me choose another. These are the cytokines, and here is the cytokine bind. This is the Jesus kinase, this is the Jesus kinase. Sorry. Now Jesus kinase phosphorylated form, it is now phosphorylated form actually phosphorylate a tyrosine residue within the phosphorylate now this tyrosine residue within the receptor. Now this phosphorylated tyrosine residue present on the receptor, cytosolic receptor, actually act as a docking site for the other enzyme like SLAT molecule. SLAT, the full form of SLAT is the signal transducer and activator of protein. Now the SLAT will bind here. Let me draw the picture again. Here is the bind, here is the P, these are the JAK and the phosphotyrosine molecule, for example here, and here the molecule called SLAT bind, and these SLAT molecules STAT. This STAT molecule actually have a SH2 domain or called Src homology domain, through which it bind with the receptor. Here it's bind. Now after the binding of these receptors, so this is the receptor, here is the solution bind, Jesus kinase, phosphorylated tyrosine molecule here and phosphorylated tyrosine molecule here, to which the enzyme the SLAT molecule bind. There are actually 6 type of SLAT molecules are present, SLAT 1 to 6, and after binding these SLAT molecules also get phosphorylated on their tyrosine molecule. This get phosphorylated on their tyrosine. Let me change the color here. Phosphorylated, also get phosphorylated here, and after that the Jesus kinase actually phosphorylate these tyrosine residues, and after that the SLAT molecules are actually dissociate from the receptor. Let me now, this is the plasma membrane, these are the receptor, recept, recept, receptor, phosphorylated molecule, but the now SLAT are dissociated from each other. But with the help of their SH2 molecule, two SLATs are now actually joined with each other to form a homodimer, and now these SLATs actually translocate into the nucleus. Now they actually translocate this SLAT, not SLAT actually, STAT. These have phosphorylated molecule. Now they actually enter, if you considering the nuclear envelope, now actually they enter nuclear, and here they actually this is the target gene of SLAT, and they actually bind here in the promoter region of their gene of target, bind with the help of some other protein. For example, this is another protein, this is another protein, it's a helper protein, and now they transcribe this region and then translate it here into protein, translated into protein. For example, this is the protein. For example, the hormone prolactin, if binds with the cytokine receptor, which I shown you in the first slide. Let me share the slide again. Here is the for example, this yellow one, if you considering as a this receptor one as a hormone prolactin, it will actually activate the SLAT, that is called 5, and actually induce the gene which produce the breast milk. This is all about the JAK-STAT pathway. Thank you.